Welcome to the Deep Dive. We grab complex sources, dig in deep, and hopefully make it all make sense for you.
That's the plan.
Today we're tackling something you might not have heard much about, but it has, well, pretty significant implications. It's a form of diabetes called Maturity-Onset Diabetes of the Young. MODY for short.
Yep, MODY. And we've got a stack of scientific articles, research papers looking into it.
Our mission for this deep dive, really unpack what MODY actually is. Why does it get missed so often? Or misdiagnosed?
And crucially, why getting that precise diagnosis is so incredibly important, both for treatment and, for your long-term health outcomes.
Yeah, get ready for some, I think, quite surprising facts. We're talking genetics, beta cells, those are the insulin makers, and why not all diabetes plays by the same set of rules? These sources make that very clear.
Definitely. So let's dive in.
Okay, where do we start?
The basics.
Let's set the stage.
Yeah.
So diabetes, broadly speaking, these sources remind us it's a chronic metabolic disease. The defining feature is high blood glucose, persistently high. And that's the problem, right? That high glucose over time.
Exactly. Left unmanaged, it leads to serious damage. We're talking heart, kidneys, eyes, nerves, major organs. It affects hundreds of millions globally. And those numbers, unfortunately, are still climbing.
It's interesting how we even classify diabetes. Historically, it was much simpler back then.
Oh, absolutely. Mid-20th century, 1965 or so. Classification was really just based on age. Infantile, young, adult, elderly diabetes. Pretty basic.
And then it shifted more towards type 1 and type 2.
Right. In the 80s, that became the main distinction. Type 1, generally seen as autoimmune, insulin-dependent from early on. And type 2, often linked with insulin resistance, lifestyle factors, maybe not needing insulin right away.
But science hasn't stood still. These sources really emphasize the picture as well. Way too simple now.
Far too simple. The last couple of decades have shown diabetes is incredibly heterogeneous. Much more diverse than just T1 and T2.
And the newer classifications reflect that.
Yeah, the 2019 WHO classification, for example, it really puts the pancreatic beta cell front and center, the cell actually making the insulin.
Okay.
And within that framework, it explicitly carves out space for other specific types of diabetes. And right there, under that umbrella, you find monogenic diabetes.
Monogenic. And that's where MODY lives.
Precisely. MODY is the classic example of monogenic diabetes.
So what does monogenic actually tell us? What does that mean?
It tells you the fundamental cause. Mono meaning single, genic meaning gene. So the condition stems from a mutation, a significant change in one single gene.
Unlike type 2, say, which involves lots of genes plus environment.
Exactly. Or type 1, which is an autoimmune attack. MODY is typically driven by that specific fault in one gene.
Okay, so one gene is the root cause. What other key features do the sources consistently point to for MODY?
All right, there are a few defining characteristics. First, the inheritance pattern. It's usually autosomal dominant.
Meaning it runs in families.
Often, yes. Strongly. You might see it in multiple generations, parent to child, and so on. But, and this is a really critical point for you listening.
Yep.
If you have young onset diabetes but don't have a family history, it doesn't automatically rule out MODY. De novo mutations can happen.
Meaning a new mutation, not inherited.
Exactly, a spontaneous mutation in that individual. So family history is a big clue, a major red flag, but its absence doesn't close the door.
That's really important. You could be the first in your family.
You could. Second key point. The underlying problem is pancreatic beta cell dysfunction. That single gene defect messes up how these cells either develop or function, how they sense glucose, or how they secrete insulin.
And the name itself, Maturity-Onset Diabetes of the Young, points to the age, right?
It does. Typically diagnosed young, usually before age 25. That's a classic feature.
And finally, how does it often look different from type 1 at diagnosis?
A key differentiator, often, is that people with MODY frequently don't need insulin right at the start. They might manage with diet or oral meds, or sometimes, depending on the type, need no treatment initially. That's quite different from classic type 1, where insulin is usually needed immediately.
Okay, let's pull that together. MODY, caused by one gene, affects beta cells, starts young, often doesn't need insulin right away, and it's pretty rare overall.
Relatively rare, yes. The sources put it at about 1 to 5% of all diabetes cases, so it's not type 1, it's not type 2, it's this distinct genetic category.
Okay. Now, here's the paradox, maybe. If it has these distinct features, why is it so often missed or misdiagnosed?
That's the challenge. The sources are clear on this. The clinical presentation, how someone actually looks when they come to the doctor, can overlap significantly with both type 1 and type 2.
Really? Like what kinds of symptoms overlap?
Well, age at diagnosis isn't always strictly under 25.
Yeah.
Sometimes it's picked up later. Things like C-peptide levels.
That measures your own insulin production.
Right. They can vary. Sometimes they're lowish, sometimes preserved. And diabetes autoantibodies, the markers for type 1. Most MODY patients are negative, but not always. Some might have low levels, confusing the picture. It's just not always clear-cut based on those initial tests.
So the symptoms aren't unique enough on their own.
Often not. And one study we looked at actually put a number on it. They estimated around 38% of MODY patients are initially given the wrong diagnosis.
Wow, 38%. That's nearly 4 out of 10 people starting off on potentially the wrong track.
Yeah.
Why is getting that diagnosis spot on so critical? What are the stakes?
The stakes are high because the correct treatment depends entirely on the specific MODY subtype, which gene is actually mutated.
Okay, so it's not just MODY, it's which type of MODY.
Exactly. Treating someone with, say, HNF1A MODY using a standard type 2 approach, or thinking they have type 1 and putting them on insulin they might not need or might need differently, it can lead to poor glucose control, unnecessary side effects, or failing to monitor for the specific long-term complications associated with their genetic type.
Okay, so the consequences can be pretty serious. What else makes it so hard to diagnose just based on clinical signs?
Adding another layer of complexity is the variability between the different MODY subtypes themselves. Someone with GCK MODY presents very differently from someone with HNF1A MODY or HNF1B MODY.
So even within MODY, it's not one size fits all.
Not at all. And even some other biomarkers that are sometimes looked at, like HSCRP, that's high-sensitivity C-reactive protein and inflammation marker, or urinary C-peptide, UC-PCR, they haven't proven reliable enough on their own to consistently distinguish MODY from type 1 or type 2, or even tell the subtypes apart reliably in all cases. One study specifically mentioned UC-PCR wasn't great for separating from type 2 in their group.
So if the clinical picture and the usual lab tests can be misleading, what is the definitive way to know if someone has MODY?
The sources all converge on one answer: genetic testing.
Makes sense if it's a genetic condition.
Right. It's the gold standard. It's the only way to definitively identify that specific single gene mutation, confirm it is MODY, and critically pinpoint which subtype it is. That unlocks the right management pathway.
We saw a study from Portugal that really kind of illustrated this whole challenge in practice, didn't it?
Yes, that was interesting. They looked at patients who met sort of the classic clinical suspicion criteria for MODY. They needed at least 2 out of 3 things: diagnosis before 25, a family history of diabetes, and signs of ongoing insulin production, like not needing insulin, having detectable C-peptide, or testing negative for those type 1 autoantibodies.
So they targeted people who looked like good candidates for MODY based on those clinical clues.
Yeah.
What happened when they did the genetic tests?
Well, they found a MODY mutation in about 50% of those suspected cases, and they even found some new mutations not previously described, which is valuable.
But only half. What about the other half who met the criteria?
Exactly. That's the key finding. A full 50% of people who clinically looked like they might have MODY turned out to be mutation negative for the common MODY genes they tested. It really highlights how difficult it is to be certain based only on clinical features, even using good criteria.
So what might be going on with those mutation-negative cases?
The researchers noted these individuals tended to be diagnosed a bit later in life compared to those who did have a mutation. They suggest some could be what are called phenocopies.
Phenocopies, meaning?
Meaning they look like MODY. Phenotypically, their observable characteristics match, but genetically, they might actually have early onset type 2, or maybe even a slow progressing form of type 1 diabetes that just happens to present in a way that mimics MODY.
Fascinating. So it looks like MODY, smells like MODY, but isn't actually MODY at the genetic level.
Precisely, which again just hammers home the need for genetic testing for a definitive answer.
This leads us perfectly into digging deeper into the specific types of MODY. The sources really zero in on a few main ones that account for most cases, right?
Yeah, they do. There's widespread agreement in the articles that mutations in just 3 genes are responsible for the vast majority, maybe up to 99% of the commonly diagnosed MODY cases.
And those are?
GCK, HNF1A, and HNF4A. Those are the big 3. Now, there are other rarer types mutations in at least 14 genes are known, but these 3, plus HNF1B, which we should definitely talk about, cover most of what clinicians encounter.
Okay, let's take them one by one. First up, GCK MODY. also called MODY-Y2. What's the story with the GCK gene?
GCK stands for glucokinase. This gene holds the instructions for making the glucokinase enzyme. Think of this enzyme as a critical glucose sensor, especially in the pancreatic beta cells. It tells the cell, hey, glucose levels are rising, time to release insulin.
Okay, so it's the trigger. What happens if that sensor gene is faulty?
In GCK MODY-Y, the mutation means the sensor is, well, less sensitive. It requires a higher level of glucose in the blood before it triggers that insulin release signal. It's like the thermostat for glucose is set permanently a bit higher than normal.
So the body just runs with slightly higher blood sugar all the time.
Pretty much. It results in mild, stable, fasting hyperglycemia, slightly elevated blood sugar. And here's the thing. It's often present from birth, but because it's mild and doesn't usually cause any symptoms, it often goes undetected for years. It might only get picked up incidentally during routine blood tests later in life. One source mentioned the median diagnosis age is around 24.
And because it's this stable, higher set point, what does that mean for treatment? This is where it gets really different, right?
Hugely different. And this is why the diagnosis is so absolutely critical, because the body's effectively maintaining its balance just at a slightly higher glucose level. Standard glucose-lowering treatments, things like metformin, other oral agents, even insulin, they're generally ineffective. They don't really lower the glucose level much, if at all.
So taking those meds doesn't really help.
Often, no. And the sources clearly state that patients who are misdiagnosed, maybe as type 1 or type 2, and put on these medications can frequently stop them entirely after getting the correct GCK-MODY diagnosis without any significant negative impact on their glucose levels.
Wow. Stopping medication could be a sign you have this type. What about the big worry with diabetes long-term complications? Eye damage, kidney damage.
This is perhaps the most striking difference. Patients with GCK-MODY have a very low risk of developing the typical microvascular and macrovascular complications we associate with diabetes, despite having this mild hyperglycemia potentially for their entire lives.
Seriously, even with higher blood sugar for decades.
Yes. The risk is significantly lower compared to type 1 and type 2 diabetes. It's a really key feature of this subtype, and another powerful reason why identifying it correctly changes everything. It avoids unnecessary treatment and provides reassurance about long-term risks.
Okay, GCK-MODY. Mild, stable, high glucose, treatment often not needed or effective, and crucially, very low complication risk. That is a world away from standard diabetes. Let's move to the next big one, HNF1A-MOY, or MOY3. This one is more common in some areas.
Yes, HNF1A-MODY seems to be the most frequently identified type in many populations, especially across Europe. It's caused by mutations in the HNF1A gene. HNF1A stands for hepatocyte nuclear factor 1 alpha.
Another gene name to remember. What does this one do?
It's a transcription factor, which basically means it's a protein that controls the activity of other genes, turning them on or off. HNF1A is important in the beta cells, but also in the liver, kidney, and gut.
So a mutation here affects how other genes work in the beta cell.
Correct. It impairs the beta cell's ability to secrete insulin effectively, especially in response to rising glucose. So unlike GCK, where the sensor is off, here the machinery for releasing insulin is faulty. There's often a progressive decline in insulin secretion over time.
Okay, so the problem is insulin release. What about treatment? Are they unresponsive like GCK?
No, quite the opposite. This is another key distinction. Patients with HNF1A MODY are typically very sensitive to a class of oral medications called sulfonylureas.
Those are older diabetes drugs, right?
They are, but they work by directly stimulating the beta cells to release more insulin. And in HNF1A MODY, they often work exceptionally well, even at very low doses, much lower than typically used in type 2 diabetes.
So getting the diagnosis means you could switch to a simple, effective pill.
Often, yes. It's a common and very beneficial treatment change that happens after a correct HNF1A MODY diagnosis. Many patients can come off insulin, for instance.
Okay, that's a big plus.
Yep.
But what about complications? If they need treatment to lower glucose, does that mean they're still at risk?
Unfortunately, yes. This is the critical difference compared to GCK-MODY pose. Patients with HNF1A MODY do have a significantly increased risk of developing the standard diabetes complications: retinopathy, nephropathy, neuropathy, cardiovascular disease.
So similar risk to type 1 or type 2.
The sources suggest the incidence is broadly similar, yes. These complications can seriously impact health and longevity. So even though treatment with sulfonylureas can be very effective at controlling glucose, careful monitoring for and management of these potential complications is absolutely essential. The underlying genetic defect still seems to make them vulnerable.
There was some mention of maybe a specific protein involved in the cardiovascular risk, apolipoprotein M.
Yeah, some research pointed towards potentially lower levels of apolipoprotein M, which plays a role in cholesterol transport, as possibly contributing to the increased cardiovascular risk in HNF1A MODY. But the exact mechanisms are still being worked out. The key message is complication risk is real and needs proactive management.
Okay. HNF1A MOEDY, faulty insulin secretion, very responsive to low-dose sulfonylureas, but carries a high risk of complications similar to T1, T2. Huge difference from GCK. Right, let's tackle the third major one, HNF4A MODY, also known as MODY1.
HNF4A MODY comes from mutations in the hepatocyte nuclear factor 4 alpha gene. Again, it's another crucial transcription factor regulating gene activity in beta cells, liver, and elsewhere. It's generally less common than GCK or HNF1A mutations, maybe 5–10% of cases in some studies.
And treatment-wise,
Yeah.
similar to HNF1A.
Yes, generally. Patients with HNF4A MODY also tend to respond well to low doses of sulfonylureas.
Is there anything particularly distinctive about how HNF4A MODY presents itself?
There is something quite fascinating, actually. The effects can sometimes manifest extremely early. We're talking at birth. At birth. How?
HNF4A mutations can cause the fetal pancreas to produce too much insulin before birth. This can lead to macrosomia, the baby being unusually large at birth.
Because insulin promotes growth.
Exactly. And sometimes, shortly after birth, these babies can experience transient neonatal hyperinsulinemic hypoglycemia, basically. Temporary but potentially dangerous low blood sugar because their pancreas is still overproducing insulin.
Wow. So a problem right at birth could actually be a clue to developing diabetes years, even decades later.
It's a remarkable link between fetal development and adult onset disease. That early history of being a large baby or having temporary low blood sugar after birth can be a significant clue for considering HNF4A NFOY later on if diabetes develops.
That's incredible. Are there other features? Does it look similar to HNF1A sometimes? Yes, there can be quite a bit of clinical overlap between HNF1A and HNF4A MODY. This isn't entirely surprising because these two transcription factors actually help regulate each other's activity. So sometimes distinguishing between them clinically before genetic testing can be tough. Although thankfully the treatment response to sulfonylureas is similar for both.
Any other associations, like with cholesterol?
Some studies have suggested a link between HNF4A MODY and dyslipidemia, particularly things like lower levels of HDL, the good cholesterol. It's another piece of the puzzle.
Okay, we've covered the big three. GCK, HNF1A, HNF4A. You also flag HNF1B MODY or MODY5 as being distinct. What makes it different?
HNF1B MODY is definitely its own beast. It's less common than the others we've discussed, caused by mutations in the HNF1B gene. Yes, another hepatocyte nuclear factor, beta this time.
Okay, what's special about HNF1B?
Like the others, it's a transcription factor, but its role is absolutely critical during embryonic development, specifically for the formation of the pancreas and the kidneys.
So it affects more than just the beta cells later in life.
Precisely. That's the hallmark of HNF1B MODY. Patients often present not just with diabetes, but with a whole constellation of issues, particularly affecting the kidneys and urinary tract.
Like what kind of kidney problems?
The sources mention things like renal cysts, multicystic kidney dysplasia, where the kidney structure is abnormal from birth uterine abnormalities in females, and often progressive chronic kidney disease. One study found kidney abnormalities in nearly half, about 44% of their HNF1B patients. Pancreatic issues are also common, like the pancreas being underdeveloped, hypoplasia, or shrinking atrophy.
So the diabetes might actually stem from the pancreas not developing properly in the first place.
That's thought to be a major factor, yes. It's not just about beta cell dysfunction appearing later, but potentially about developmental defects impacting the organ structure itself.
What about the usual diabetes complications like eye or nerve damage?
They can still occur. Retinopathy and neuropathy are reported, but really the major clinical concern and the dominant feature driving morbidity in HNF1B MODY is often the kidney disease. Seeing significant kidney or pancreatic structural problems alongside diabetes in a young person should set off alarm bells for testing specifically for HNF1B mutations.
It really paints a picture of how diverse MODY is. Understanding these specifics seems crucial, but also challenging to study. How are researchers actually investigating the mechanisms?
You're right. Understanding the exact pathology, the step-by-step process of how the mutation leads to the disease is tough. Getting hold of human pancreatic tissue for study is obviously very difficult.
And animal models don't always work well.
Often not. Standard lab mice, for example, frequently don't replicate the human MODY phenotypes accurately. HNF1B might be a slight exception, but for many types, the mouse models just aren't quite right.
So how do scientists bridge that gap? What are the newer approaches?
This is where things get really exciting technologically. Researchers are increasingly turning to human-induced pluripotent stem cells, HIPSCs.
Those are adult cells reprogrammed back to an embryonic-like state.
Exactly. You can take a skin cell or a blood cell, reprogram it, and then guide it to develop into almost any cell type in the body, including pancreatic beta cells right there in a dish.
So you can make beta cells from a MODY patient in the lab.
Precisely. Or you can take cells from a healthy person and use gene editing tools like CRISPR-Cas9 to introduce a specific MODY mutation.
CRISPR the gene editor.
Right. This allows scientists to create what are called isogenic cell lines. That means the cell lines are genetically identical except for that one specific MODY gene mutation they're interested in.
Why is that so powerful?
Because it removes all the background genetic noise. You know that everyone's genetic makeup is slightly different? By comparing a cell line with the mutation to its identical counterpart without the mutation, or with the mutation corrected, researchers can directly pinpoint the specific effects of that one mutation on cell function, development, insulin secretion, whatever they're studying. It gives a much clearer cause and effect picture.
Are these HIPSC models actually teaching us new things about how MODY works at the cellular level?
Absolutely. The sources highlight several examples. Studies using these models have shown how HNF1B mutations mess up the development of the early pancreatic progenitor cells. They've provided evidence for HNF4A mutations causing the protein to end up in the wrong part of the cell.
And for HNF1A?
For HNF1A, these models are revealing detailed defects not just in the final step of insulin secretion, but also upstream in how the cells process glucose through glycolysis and how their mitochondria, the cell's powerhouses, function. And for some rarer types, like those caused by KCNJ11 mutations, they're showing exactly how defects in ion channels disrupt insulin release.
So this basic science, using these sophisticated tools, is directly translating into a deeper understanding of the disease mechanisms.
It is. And that fundamental understanding is absolutely crucial if we want to move beyond the current treatments, like sulfonylureas, and develop new therapies that target the specific underlying problems in each MODY subtype. That's the long-term goal.
We touched on this earlier, but the sources also give a bit of a global perspective. Does the prevalence of these types vary much around the world?
It does seem to vary. While GCK, HNF1A, and HNF4A are generally the most common culprits worldwide, their relative ranking can flip depending on the country or population being studied.
Like HNF1A being more common in Northern Europe?
Right, like in the UK or Norway, whereas GCT seems to be more frequently found in places like Portugal, Italy, and also notably in Japan. So there are definite geographical differences in the genetic landscape of MODY.
And beyond the big 3 or 4, the others are much rarer.
Much rarer. As we said, mutations in at least 14 genes are implicated, but GCK, HNF1, HNF4A, and HNF1B together account for the lion's share of known cases. All the other subtypes caused by mutations in genes like NUROD1, KLF11, PECX4, INS, etc. are individually very rare, collectively making up probably less than 5% of diagnosed MODY.
Are there regions where maybe even the common types are less common, suggesting other factors are involved?
That's an interesting point raised in some sources. Studies from some populations, particularly in parts of the Middle East, have sometimes reported finding fewer cases caused by the typical HNF1A or GCK mutations than you might expect based on European data, even among patients clinically suspected of having MODY.
What might explain that?
It could suggest that perhaps other, less well-characterized genes play a larger role in those populations, or maybe different inheritance patterns are more common. Factors like higher rates of consanguinity marriage between relatives could also influence the prevalence of certain genetic conditions, particularly recessive ones, although most common MODY types are dominant. It really just underscores that our understanding of the full genetic spectrum of MODY across the globe is still evolving.
Okay. So after this deep dive, what's the big picture? What are the main takeaways from these sources about MODY?
I think the main takeaway is that MODY is this distinct, fascinating, and often hidden subtype of diabetes. It's fundamentally genetic, caused by that single gene defect. It typically starts young, often runs in families. But the absolute key is that the specific gene involved changes everything.
Everything? Like what specifically?
Like the clinical presentation, the severity, whether you need treatment, what treatment works best, and perhaps most critically, your long-term risk of developing serious complications. It all hinges on that specific gene mutation.
And because the symptoms can easily be mistaken for type 1 or type 2, misdiagnosis is a real problem.
A significant problem, yeah. And as we've discussed, getting that diagnosis wrong can have major consequences for health management, leading to ineffective or unnecessary treatments and inadequate monitoring for potential risks.
Which makes accurate genetic testing not just useful, but often essential for proper care.
Absolutely. It's the key to personalized management for these patients. And looking forward, the advanced research using stem cells and gene editing holds incredible promise for really understanding these diseases at a molecular level and hopefully developing even better, more targeted therapies down the road.
So for you listening, if you or someone in your family was diagnosed with diabetes young, maybe before couple 5 or 30, or if there's a strong family history of diabetes across multiple generations, especially if typical type 1 or type 2 features don't quite fit.
It's definitely worth having a conversation with your doctor about the possibility of MODA. Asking whether genetic testing might be appropriate could be a really important step towards getting the clearest possible diagnosis and the most effective care plan.
Okay, let's wrap up with a final thought for you to chew on. We've seen these huge differences between MODY subtypes, right? Particularly the stark contrast in complication risk between GCK-MODY, where risks are low, and HNF1A-MODY, where they're high, similar to type 1 or 2.
A massive difference in prognosis.
So here's the provocative question. If genetic testing for MODY became much more widespread, maybe even a routine part of the diagnostic process for all young people presenting with diabetes, how might that fundamentally shift how we even define and classify diabetes? Would we start thinking about diabetes primarily through a genetic lens first, especially in younger individuals, rather than relying so heavily on the initial clinical picture or age alone? What would that mean for treatment pathways and public health approaches globally?
That's a big question. Would genetics become the primary classifier, overshadowing the old type 1, type 2 split in many cases? Something to think about.